Okay, this is going to be lecture 7 review this is a review of the hormone receptors. Okay, so got my notes. This is the entire lecture 7. We started off the lecture talking about the lock and key analogy between the hormones are the key and the receptors are the lock that the key unlocks, opens, activates. So hormones, when they bind to receptors, their nomenclature changed. They become known as ligands. Ligands are what bond to receptors. Hormones become known as ligands when they bond to the receptors. Receptors are made of proteins. There are different kinds of receptors. You have transmembrane receptors, intracellular receptors, and extracellular receptors. There's 3 domains of these receptors. And receptors are highly localized. You have different receptors on different organs. You have sometimes different receptors in different parts of the body, depending on where it is in the body, not only what organ it is, but just where it is. For example, you have receptors on many different kinds. You have kinds of serotonin receptors in your gut, and you have serotonin receptors in your brain, but those receptors do different things. For example, in your brain, the sort of secondary messenger cascade that happens when serotonin bonds to those receptors causes you to feel happy, or stabilizes your mood so that you have more of a normal or regular mood. In your intestines, it inhibits water absorption in your intestines. So then if you have too much serotonin, then you'll have diarrhea, for example. It's just one example. There are probably thousands. When we're talking about receptors and we talk about a receptor's affinity for something, we're talking about how tightly the hormone, the ligand, bonds or binds to that receptor. So a high affinity means it binds very tightly, and it's very hard for anything to remove, or maybe the receptor just won't let go of that hormone, of the ligand. Let's see. They usually have a low capacity. It's usually like a 1-to-1 ratio for receptors. 1 hormone for 1 receptor. Or even less. Sometimes it takes 2 receptors for 1 hormone. Those are dimers. It's when we talk about dimerization. Where was I? There's usually a low concentration of receptors. There's not many of them. They're also dynamically regulated, so if you have too much of not of the receptor, but of the hormone, your body genomically, your body will code for less of that receptor because of the abundance of the hormone. You don't want it's to regulate the not only the expression but the effect that hormone has. That's how you regulate. One of the ways your body regulates. You can also have a desensitization of a receptor, so it becomes its affinity basically changes. It becomes a higher affinity or a lower affinity depending on the concentration of the hormone for that effect. Let's see. Concentration. They're highly selective, so receptors will only take certain structures, and usually what that translates to is different species of hormones have a complementary receptor. So it's usually 1-to-1. Not usually. Anything that looks similar to the hormone will bind to that same receptor. The effect might be a little different because these are organic compounds. So even if the mold, if you will, if the key is not the exact shape, there's only like a little difference, a little twinge, it can still affect that receptor. It just might have a lesser effect. Or no effect. It might just block that lock. Highly selective, similar molecules. Again, on the topic of affinity, how tightly it binds. Binding is reversible, so the receptor can let go of a hormone and it goes back in. And then sometimes that hormone is recycled, sometimes it's broken down, etc., etc. It's binding. Okay, so receptors are dynamically regulated, as I said before. There's many different mechanisms of action for receptors, and the functions of each receptor varies. More on that later. Until a receptor is considered free until a ligand binds to it. So it's an unoccupied receptor. When a ligand binds to it, it is then referred to as a occupied receptor. That's it. Receptors that are found on the cell membrane for hormones that don't cross the lipid bilayer. We want to say the bilipid layer. So like peptides, proteins, catecholamines, receptors for these cell types have receptors on the surface of a cell because they cannot enter the cell. So that's where you get like your transmembrane or even just your extracellular membrane receptors. And then you have intracellular receptors for when the hormone can, in fact, enter the cell, like steroids and thyroid hormones. These cell membrane receptors, right, I just talked about that. So it binds the receptor on the outside of the membrane, and that triggers an intracellular response, which in turn usually triggers secondary messengers, like cyclic AMP is the big one, but it can be other things as well, like ions, for example, or just kinases. So we're going to briefly go through the domains. The first domain we're covering is the transmembrane domain, which predominantly, with respect to what we're talking about in this class, we're talking about insulin, growth hormone, and then with growth hormone, the genus kinase and the STAT molecules. So there's different kinds of transmembrane receptors. You have the single domain, or not domain, the single transmembrane receptor, which only crosses the membrane once. So it's basically you have the membrane, and then the receptor is just, you have extracellular, it's a receptor, and then an intracellular component. Goes all the way through the membrane, that's why it's transmembrane. You can also have a 7-transmembrane receptor, also known as a GPCR. It intersects the membrane 7 times, but we're going to talk about that later. So the single transmembrane domain is also called a tyrosine kinase receptor, generally for insulin, growth hormone, and prolactin. To understand this, you kind of have to know what a kinase is, and kinase is basically any sort of kinase is some sort of phosphorylating enzyme that converts ATP to ADP with an organic phosphate for free energy, if you will. So the insulin receptor, this is where we talk about dimerization. Where is this? The growth hormone. No, it's yeah, it's insulin. So for insulin, you have dimerization. It requires it's a 1-to-1 ratio. It requires 2 molecules of insulin. They each bind to the alpha subunit of the transmembrane receptor. The binding of these two insulin to the subunit causes them to come together. They have an affinity for each other, come together. And when they come together, you get, they dimerize, that's what it's called. So if you only have one bound, nothing happens. You have to have both. So when both are bound, they come together, and then you have this, it's called autophosphorylation. So you have 3 or 4 phosphates at the base or at the intracellular component of the single transmembrane receptor, and that so you have the autophosphorylation, which then stimulates the glucose transport protein, like the formation of and the movement of into the cell membrane, and the GTP. It's shorthand GTP. It's not the guanosine triphosphate. It's glucose transport protein in this instance. And the glucose transport protein does as what you'd expect by the name. It transports glucose from the extracellularly to the inside of the cell. It allows for the inward flow of glucose for the cell, and that's what insulin, right? Because it's your sugar regulation. So you have your insulin binds to the receptor. You have a series of intracellular effects, and then it allows the cell to take in glucose from the bloodstream or from like the surrounding area, so that one, you don't have as much glucose running around in your blood, which can cause negative effects, and also 2, allows the cell to have glucose that it can then break down for energy and to survive. The next example we're talking about is growth hormone, GH. For this one's a little bit different than insulin. So you have one growth hormone binds to 2 receptors. The ratio is no longer 1-to-1. It's 1 hormone for 2 receptors. And it binds this, and it makes a when the growth hormone binds to the 2 receptors, it forms what we call a Janus kinase complex. Yeah, that's the JAK, JAK. And from here on forward, I'm going to refer to the Janus complex as JAK, just because it's easier. So growth hormone binds to the 2 receptors, so you have the activation of the receptors, and they bind to intracellular JAK proteins, and the JAK proteins intracellularly become phosphorylated, and then those phosphorylated JAK proteins cause, in turn, the signal transducer and activator of transcription, or STAT proteins, to become phosphorylated. So you have the phosphorylation of the JAK proteins, in turn catalyze the phosphorylation of STAT proteins. And then the STAT, which is a transcription factor, it's a TF, it causes genomic effects. And what does that mean? So, is that all I read? So, it's growth hormone, and if you remember you should remember what growth hormone does. It causes the growth of things. It causes your body to release like proteins. It causes protein synthesis. In general, it causes the growth of things, of organs, of tissues, of cells. It can have other effects on diet and testosterone release. For example, And then from there, we're not going to talk about prolactin right now. We'll talk about that when we get to parturition, which is Thursday, or later this week, later in this semester. So that's really all we talked about for the single transmembrane domain. Now for the 7 transmembrane domain, you should know that they're called GPCRs. They're about 400 to 600 amino acids long. The chain crosses 7 times over the membrane, and it's made of an alpha, a beta, and a gamma subunit. The alpha subunits are the enzymes of the GPCR. So you have 4 different kinds with their denominations, one being S standing for stimulatory, and it stimulates adenylate cyclase, and then you have an inhibitory one, which inhibits adenylate cyclase, and then you have Q, which causes PLC, which stimulates phospholipase C, and then you have T, which stimulates GMP, phosphodiesterase, cyclic GMP, which cleans up adenylates, cyclic GMP, basically, which we're also not really going to talk about. So I've just mentioned the important thing to remember here is you have an alpha subunit that has a stimulatory and an inhibitory effect on adenylate cyclase, which becomes important soon or now. So what is adenylate cyclase? Adenylate cyclase is this secondary messenger. It is an enzyme that converts ATP to cyclic AMP. And cyclic AMP is a very powerful and very widely used secondary messenger. It's very short-lived, and it's inactivated by phosphodiesterase. Not cGMP phosphate, but cyclic AMP phosphodiesterase. There are 2 different species of molecules. Cyclic AMP activates PKA, which is part of the cascade. And the end result of PKA, when epinephrine triggered PKA, the epinephrine cascade triggers the release of glucose into the bloodstream. It's a non-genomic effect. And in general, PKA can have both non-genomic and genomic effects. For example, convenient list, epinephrine can trigger, so we're going to talk about the hormone epinephrine, or the neurotransmitter. It's kind of both. The non-genomic folks of epinephrine. So in the liver and the muscle, it stimulates glucose release into the bloodstream so that your muscles have glucose to break down, and then allows you to, exercise, to run, to panic, run around in circles. In your adipose tissue, epinephrine activates hormone-sensitive lipase, or HSL. And HSL is really cool because it stimulates lipolysis, so a breakdown of fat. And when you have the breakdown of fat, you then have the components needed to make hormones or to make more glucose. And that's great. And epinephrine also, as you've probably experienced at some point in your life, or many times in your life, on your cardiac muscle, it activates, I have to read this. What is that? Phospholambin. So phospholambin binds to calcium bonds and allows for the influx of calcium into your cardiac cells, which, as you should know from previous classes, calcium is important for the contraction of muscles. the genomic effects of epinephrine, of PKA, phosphokinase A, phosphorylates Krebs. And if you should remember this from like biochemistry or pretty much any lower level biology class, Krebs is a very important transcription factor. It is used, we have something called, not to be confused with the Krebs cycle. That's spelled with a K, this is a C. So it's a transcription factor, and that causes really protein synthesis. And I'm going to be very vague about this now, but we'll talk more about why Krebs is important and what it does in the body in relation to hormones at a later time. But for now, you should just know that it causes slow and long-lasting effects. An example of this is if you have epinephrine being released in your body for a long period of time, that basically means you're like chronically stressed or you're chronically in danger. And at a long period of time, that can be really detrimental. A short period, it can be really beneficial. And if you've taken endocrinology before, and you're just using this to review, you should know that we operate every day. Our body operates in such a way. That's a poor way of setting it up. We function through homeostasis, right? We have negative feedback loops to regulate our hormones and proteins and pretty much body temperature, hypertension, pretty much everything in the body is negative feedback regulated. And if you're under chronic stress, you are attaining allostasis. Your body is trying to move from its current homeostatic state to another homeostatic state that has a higher epinephrine release in it. However, if this might be going beyond the scope. If that is too chronic, and the amount of stress you're on keeps increasing, your body never reaches that allostasis. And you reach what is known as an allostatic load. So it's the maximum capacity for change your body can have in a short period of time. And when that's bypassed, your body starts to break down. You start having higher rates of mutation, which they're usually bad mutations, and they eventually lead to your death. Your immune system becomes compromised. Muscle tissues start atrophying, start to atrophy, and you're gonna have a bad time. Anyway, so now we're going to talk about G proteins and phospholipase C, or PLC, which is another important thing. Eventually, it can trigger genomic effects, doesn't always. So PLC is activated by G proteins, and it's really important for signal transduction. I get more on that later. PLC cleaves PIP2 into DAG and IP3. I'm not going to go, it's like phosphoinositol something. Can't remember at the top of my head. DAG is diaglycerol. Easy. IP3 is inositol phosphorus. They're long names. Just remember PIP, subtext 2, and then DAG and IP3. So PLC, phospholipase C, cleaves PIP2 into two components: DAG, which stays in the membrane, and IP3, which goes on to become a second messenger. And actually, through a series of effects, I think eventually leads to cyclic AMP, and then you have cascade. Anyway, both DAG and IP3 are second messengers. They're just intracellular second messengers. DAG never leaves the cell membrane though. That's important to know. So IP3 eventually gets down to the endoplasmic reticulum, and basically the end game of PLC is to increase the amount of calcium within the cell. So when IP3 and the following steps get to the endoplasmic reticulum, the ER, it triggers the release of calcium from the ER into the cytoplasm. And basically the increase of the concentration of calcium in the cytoplasm can then itself triggers other things, and that calcium can eventually be used for things like contractions, general mobility, your metabolism, exocytosis, eventually going through the Golgi apparatus. It can be an inflammatory response, and is also very important for your memory. I skipped a part. In relation to calmodulins, which calmodulins are receptors that are, 1, highly conserved, 2, they're expressed in most cells. Like, it's a highly conserved receptor, like genetically, and it's in most animals. It's everywhere. It's only found in cytoplasm. It takes one calmodulin receptor takes 4 calcium ions. That's also important to know. It's a 1-to-4 ratio. And then you should also know that G proteins, GPCRs, can also have effects on ion channels, and the common ion channels are, of course, potassium, sodium, chlorine, and calcium. It can affect the opening and closing of these channels, which can in turn affect nerves and muscles and all of your organs, and signal transduction, of course. Anyway, that's it. That's it for lecture 7. If you have any questions, please ask. I need to study this, just like you do. There's a pretty good book. Whatever, endocrinology. This is, what edition is this? I have no idea. 5th edition. It was like $80. This year, I don't need it. I'm still gonna use it. It's nice. It's a good read. It's fairly well presented. You know. Yeah. So I'll see you next time. Actually, you'll probably see like 5 of these being uploaded today. But yeah. Peace out.